Today I'm going to present posters that I will present at the ISMB conference in Lyon in July. This poster is about how protein structure dynamic improve catalytic site prediction. So I am Aubailly, and this poster has also been made by Digrongruo Yu and Christian Roach. So within the last decade, great efforts have been made to find catalytic sites in enzyme. However, due to the complexity of experimental method and with the rise of machine learning, prediction algorithms based on 3D structure were developed. But while many models took into account features on the static 3D structures, to our knowledge, none of these have used dynamic feature extracted from an elastic network model of the protein structure. So the idea of this study is to combine structural parameter, normal mode of vibration, chemical feature, and sequence information to predict catalytic sites in enzyme. Well, in the method section, I will present how do we describe each feature. Then I will present the results and conclude. So for the method, as we can see, each protein residue is described in 4 groups of features: biological, chemical, dynamic, and structural features. To start with the biological feature, we compute from the PSSM matrix the Shannon entropy, the maximum percentage occurrence, and the number of different observed amino acids for each position. Concerning the chemical feature, this is a one-hot encoded frame where each residue is labeled as polar, charged, or hydrophobic. Concerning dynamic feature and structural feature, they are both extracted using the coarse-grained framework and the anisotropic network model. The coarse-grained framework with the anisotropic network model consists of describing a protein with beads and spring. Each amino acid is a bead that is connected to the other amino acid in its surrounding. Typically, there is a cutoff radius RC, and when the distance is less than this cutoff radius, 2 amino acids are connected. So this connection of amino acids allows us to describe a protein and map it like it could be a graph. So this is the structural features. Based on those graphs, we can compute the degree centrality, the closeness centrality, and the betweenness centrality. Concerning the dynamic feature, by using the potential energy, we can extract all the mode of vibration by diagonalizing the Hessian matrix. And from this, we can extract the 10 slowest mode of vibration and the 10 highest mode of vibration. Then, what we have done, we have combined all those features for each amino acid, and we have trained a logistic regression on the catalytic site atlas database. These training and testing sets correspond of 564 proteins for the training and 126 proteins for the testing dataset. And here on the left is an example of how it looks like for a specific protein. This is the protein alcohol dehydrogenase E chain, PDB code 1QLH. We have on the x-axis the residue number and on the y-axis the probability of being catalytic or not. The annotated catalytic site from the catalytic site atlas are marked as black circle here. And as we observe, there is a good correlation between high probability and catalytic sites. If we want to deep dive about what are the feature importance for the logistic regression to predict whether a residue would be catalytic or not, we have the feature importance here, and it turns out that the 5 most important features concern biological, structural, and chemical features. However, we have observed that although the dynamic features do not contribute a lot to the prediction when we observe this graph, it turns out when we remove them, it drops significantly the recall metrics. And as a general statistic point of view, we have observed on all the test sets a ROC AUC of 0.95 and a precision recall of 0.17, 0.78. So to conclude, we present here a new model to predict catalytic sites in protein. Our model includes all kinds of features, biological, chemical, structural, and dynamic features. And the idea is to improve catalytic site prediction. What we have observed is that when we remove one of those features, it dropped significantly the accuracy metrics, underlying the fact that we need to consider a protein as a global system. What we have achieved is a model with a good accuracy compared to previous published method. In the next step, we would like to fine-tune the model or test new model. For example, for now it is just a logistic regression, but we can improve the prediction algorithm. Moreover, the idea would be to test different use cases for the model to better understand its applicability. Is the model able to better predict catalytic sites in subgroup of enzymes, for example, sharing similar characteristics? This is a question we would like to answer. And all this work wouldn't have been possible with in vitro cell research that I would like to thank. Thank you for your attention.